seen over all these? Focused on these now, right? Those are the ones we did in lab this week. You guys are all learning this, right? So that, like when I say which one is a protostome, right? Protostome means it's one of these guys, right? And so I basically gave you two cnidarians, a sponge, and one of these, right? And that question you just had a minute ago? Yeah. You're starting to organize all these phyla into these groups, right? So that if I ask you, is this a protostome or a deuterostome, is it a bilaterian or not, right? You can sort of place it in there. And this is the slightly different tree that's probably a little bit more accurate, right? Showing sponges not as a monophyletic group, but as a basal grade, paraphyletic grade in animals. Then ctenophores and cnidarians probably not really actually going together to form a monophyletic group. Aseals being pulled out of platyhelminths so that they're potentially the sister taxon to the rest of the bilaterians. These would be then the protostomes, and these would be the deuterostomes, with the platyhelminths being the majority of them, like the ones I was just showing you being within the Lophotrochozoans. And actually, the simplicity of their bodies probably being a reduction or a derived condition rather than the ancestral condition, as was historically thought. That's why in that movie, did you notice how they're saying they're the first ones to have sex, and they're the first ones to find food, and all that kind of stuff? It's because they're still talking from the old thinking that platyhelminths occupy a basal position within animal phylogeny. That's an outdated aspect to this movie. Yeah. Obviously, if they're a derived group, they're not the first to do anything. Okay, so this shows you sort of which group we're talking about now. Protostomes that are Lophotrochozoans. This is what lab this week was supposed to be. This is a, did you guys see this? A couple of you did that were there last night. Who had this? Oh, I can't think who had this now. Whoever had lab yesterday, there was somebody that, it was Lauren actually, the other one. She had the elosoma, and she put it on dark field so you'd see the little CT just beautifully, and that little elosoma was just teeming with rotifers. They had their little heads and they were just spinning like this. They were the coolest little things. I didn't see them the Tuesday night thing, but we should look. If you remind me next week, that elosoma will still be up there. I should make a little mount. We're not covering rotifers really though, but they were there yesterday. They were in with the elosoma. These are bryozoans, brachiopods, and nemertians. We're not really covering any of those. So the Lophotrochozoans that you really need to know are platyhelminths, right, which we just finished. And then annelids. Who's this? Here he is. And here's a little Christmas tree worm, right? And mollusks, which would be things like cephalopods, gastropods, and bivalves, right? So here we go. What makes an annelid an annelid? The common name for this group, like platyhelminths, are flatworms. The common name for annelids are segmented worms. And in fact, the name anillo in Spanish would be a what? A ring, right? So they have little rings, little segments. And I think I mentioned, I'm sure I did before, for a very long time, people thought that these were closely related to arthropods because arthropods are also segmented. It ends up that the segmentation you see in arthropods and annelids is convergent. They're not closely related to one another, but these guys definitely are segmented. So they're bilaterally symmetrical. Everybody from here on out is bilaterally symmetrical, except for echinoderm starfish and sea urchins are radially symmetrical as adults. They're pentaradially symmetrical, but they're still bilaterians. Everybody's a bilaterian from now till the end of animals that we're talking about. So they're bilaterally symmetrical. They're triploblastic. Everybody's going to be triploblastic, which means what? They have mesoderm, 3 germ layers, right? They have spiral cleavage. Who else just had spiral cleavage a minute ago? Platyhelminths. And the other group that's going to have it is mollusks. So if I ask you what's a synapomorph you could potentially use for Lophotrochozoans, spiral determinate cleavage would be one, right? We find it within only that group. Now there are other Lophotrochozoans like rotifers that have a pseudocoel. They don't have they have, sorry, on the wrong character. That have radial cleavage, right? That don't have determinate cleavage the way that you find in these. But apparently this particular type of determinate cleavage might have evolved on this branch leading to annelid mollusk helminths. Okay, they have an open gut, a mouth and manus. Again, that's going to be true for everybody going forward. They're protostomes, meaning the blastopore becomes the mouth. And they have a schistosele, which is a trusolome that arises how? From a, what does schizo mean? Schizo? If you're schizophrenic, you have a split personality. So a splitting of mesoderm, right? That's what gives rise to the schistosele. And they have this special type of a larva that I'll show you in just a second called a trochophore larva with the little ciliated, apical ciliated tufts, right? When I show you mollusks, you'll see that the first 3 lines of this I just went copy, paste. All of those things are true for mollusks too, right? There's segmented worms, little rings is what Annelida means, right? They're not just ringed on the outside, but they're ringed on the inside too. So an earthworm is actually septate. Where'd you hear septa before? Remember that? In the fungi, remember the little cross walls in the hyphae, right? So there's like little walls that are in there that actually divide the worm into segments internally as well as externally, right? They have both circular and longitudinal muscles, right? You saw that yesterday in your earthworm that you could get short and fat, long and skinny. Anybody who can do that scrunchy thing has got both sets. You couldn't do that otherwise. You'll see this will be a good distinction from nematodes. Nematodes have only longitudinal muscles, so how do they move? Serpentine, side to side, they can thrash like this, but they can't scrunch, no scrunching, if you don't have circular and longitudinal muscles both. Here's an interesting one about annelids. Annelids have what's called a closed circulatory system. So what that means is that all the blood in their body is completely contained inside of blood vessels, like what you would consider capillaries or veins or arteries or something. That probably doesn't sound like a big deal to you because you have a closed circulatory system, right? Vertebrate animals have closed circulatory systems, but it's unusual in invertebrate. So for example, if you looked at something like a grasshopper or a cricket, it has an open circulatory system. There's a little dorsal blood vessel, and it basically pulses. Blood is drawn in one side, spewing out the other, and then the blood, most of it, is just bathing this giant cavity inside of the little insect called a hemocoel. But the blood is not contained inside of little blood vessels the way it is here. Yours definitely is. In fact, if you hit yourself and break blood vessels and the blood comes out of the blood vessels, you get a big bruise, right? I mean, your blood is completely inside of blood vessels, inside of arteries and veins and capillaries, right? You don't have blood that's just floating around inside you. If you did, we say that you are bleeding internally. That's not a good thing, right? Your blood is all contained in blood vessels. So is an earthworm. And earthworms is too, but that's unusual for an invertebrate animal. 3 classes, right, that you're learning. This is the phylum Annelida, so we can divide it into classes. You looked at these in lab, right? Polychaetes, oligochaetes, and Hirudinates. So polychaetes have these little feet-like structures called parapodia, with these long little bristles called setae. And your poster child polychaete is Nereus, right? That's the little one that was up in that bowl. Oligochaetes have setae, but they're a lot shorter, right? Hopefully you felt them when you rubbed your little earthworm from tail to head on his ventral side, right? And they have this thing called a clitellum, which is that little structure that secretes mucus and sticks them together when they copulate. Hirudinans are going to be leeches, and these are characterized by having anterior and posterior suckers. Some polychaetes, a few of the polychaetes have separate sexes, but these guys are, oligochaetes and hirudinans are all hermaphroditic. So earthworms are both male and female, right? The leeches up there are both male and female. Yeah. This shows you an open versus a closed circulatory system. So most invertebrate animals would have something like this, where there's a dorsal blood vessel that pumps, it draws water in, draws blood in through these pores and then spews it out here, but most of the blood is just loose in the body. You know how like if you're driving down the road and an insect hits your windshield like that, it just everywhere? Yeah. Like if you had a mouse hit your windshield or something, it wouldn't do that, right? It would just splatter like that unless it hit really, really hard. But an insect, the blood is just loose inside of the insect. It's not contained inside of blood vessels the way that it would be in something like an earthworm. So an earthworm, you'll notice, actually has a dorsal and ventral blood vessel, and it has these little hearts, which aren't real hearts. They're just little tubes that pulse, right? But they're not really chambered hearts the way that we think of them in a vertebrate. But all the blood that's inside of the earthworm is completely contained inside of blood vessels, so we say that the system is closed. The circulatory system is closed. This is a good point to get you to start thinking about something here. You'll notice that the circulatory system, earthworm is a little different because it has a closed system. But in this cricket here, the circulatory system, the blood vessel that it has is running along what side of the animal? It's on the dorsal side. Where is your circulatory system though? It's ventral, right? Like if I wanted to operate on your heart, I wouldn't go in on this side. I'd go in on this side, right? Your circulatory system is here. You'll see in a moment, these guys actually have ventral nerve cords. So their nerve cords run along this side. These guys also have ventral nerve cords. So their nervous system is ventral. Where is your nervous system? It's dorsal, right? So if I wanted to operate on your spinal cord, for example, I wouldn't go in this way. I'd go in this way. In virtually all invertebrates, the nervous system is ventral, the circulatory system is dorsal. In vertebrate animals, the nervous system is dorsal and the circulatory system is ventral. So there's this hypothesis called dorsal ventral axis inversion. You know what that means? It means that the idea is that at the base of vertebrates, that basically what was dorsal became ventral, and what was ventral became dorsal. So what your ventral side in an invertebrate animal really would be its dorsal side. Yeah, remember this. I'm going to mention it again, but I want to point it out to you right now so you can start thinking about it. This shows you what a trophophore larva looks like. There's it is in a cartoon. This is what a little baby worm, like those little nereid worms, the marine worms, they go through a larval stage like this. And if I show you a trophophore larva for like a marine snail, guess what? It looks a lot like this. Remember how we were focused on this idea that annelids and arthropods were each other's closest relatives? There's no arthropod that makes any kind of a larva like this at all. We're going to look at lots of arthropod larvae. There's never anything like a trophophore larva, but a snail makes a larval form very much like this, supporting this idea that annelids and mollusks are actually more closely related to each other, right? Not arthropods. This shows you a cross-section through who do you think? Here's the little parapodium, right? The CT. This is Nereus, right? A little cross-section through Nereus. And you'll notice there's a true coelom, there's longitudinal muscle, circular muscle. This is all mesodermal tissue, right? So that coelomic cavity is completely lined with mesodermal tissue. There's a ventral blood vessel and a dorsal blood vessel because it has a closed circulatory system. Here's the gut. I put this on exams before. I'll copy, paste, and I'll go, is this a platyhelminth, nematode, or an annelid? You'd all write annelid, if you remember. And then I'll say, how do you know? How would you know, based on what's labeled there? One way you might know is it has both longitudinal and circular muscles. Can't be a nematode. Nematodes don't have circular muscles. Has a true coelom. Nematode's going to have a pseudocoel. A platyhelminth has no coelomic cavity whatsoever. It has these both this dorsal and ventral blood vessel, indicating sort of a closed circulatory system. Again, you wouldn't notice that. And of course, things like Parapodia and CT are annelid characters. You don't find those in nematodes or platyhelminths. Would you all get that right? Maybe, sort of, kind of. Here it shows you through an earthworm. It's kind of the same thing. Here they actually show the little CT, the little bristles that you were feeling when you were rubbing your earthworm. You'll notice you've got the closed circulatory system here with both the dorsal and ventral blood vessels. Here, like I just said, there's the ventral nerve cords. Notice its nervous system is ventral, not dorsal. And it's got some relatively sophisticated little excretory organs. You also see the septa that divide the worm internally. And you can see here's the intestinal tract, and then you also have the coelomic cavity here, and both are they able to show both longitudinal and circular muscles? Not too complicated, but you're going to learn a little bit about the anatomy, right, for these guys. So what do you think happened in there? Yeah, they're copulating. This is the clitellum, of course, is secreting this mucus that actually facilitates this. My understanding of earthworms is that they don't really worry about who's going to be the mom and who's going to be the dad. They kind of do both. So like one inseminates, they inseminate each other, and then they each go off and have kids. So they kind of share.
The fetal thing, you don't have segmentation on it?
It is segmented, but it's thickened, I think, to the extent, I don't, so you're asking, is this a single segment here? I don't think so. I think there's actually multiple segments, but we should look that up to make sure. But I just think it's so thick on the outside, the epidermal layer, that you don't actually see where the segments are.
That's the clitellum, right?
Is what?
What did you call it?
Clitellum. Yeah, clitellum. There's a word used to describe oligochaetes and hirudinans. It's called the clitellata. So polychaetes don't have clitellum at all there, but you do find them within the oligochaetes and hirudinans. Okay, so these are the three classes you're learning, right? Oligochaeta, Polychaeta, and Hirudinea. And at least for these two, you have some that you know their name, right? So notice these guys have a reduced head and they don't have parapodia. You know four. What are the four you know by name? Lumbricus. Come on, guys, they were live and laugh. Tubifex. Y'all looked at Tubifex, right? Those little black worms. Darrow. Did you all see Darrow? And Elosoma. Right? You recognize those four, right? Polychaete, who's your little poster child for that? And notice these, Nereus, right? And notice these guys have a well-developed head, they can have little antennae, little eyes and stuff, so more kind of millipede looking really than these guys would. And the Hirudinaeans are the leeches. There you don't have to have a specific genus name, huh?
What's the key? Like that word?
Chaete are the
Yes.
chaete or the setae are the bristles. The bristles.
Oh.
So this shows you a typical polychaete, right? With these little setae, here they are, the bristles, and they have the little feet-like things, the little parapodia. Here's Nereus here. This is another errant polychaete. So a lot of these polychaetes you'll see actually live inside of tubes. This is an errant one, meaning he's out kind of like running around. The common name for this is a sea mouse. It looks like a little mouse. The setae, look at how beautiful these bristles are. They're so long that it almost looks like he has hair or something. And you'll see some beautiful fireworms at Moat when we go, and they have very long setae too that actually have venom in them. So if you touch one of those, you get a really bad sting from it. Obviously Nereus doesn't, because
Yeah, I actually saw the little, almost look like a vein going down too.
Yeah.
The branches coming out or something.
yeah, right. Well, and you can also see that there's a very prominent dorsal blood vessel that you can see on the back side. Did you notice that these guys are really shiny in the back, the Nereus? They have these collagen fibers that are interwoven that give them that iridescence on the back side. And you can see the dorsal blood vessel too. And there's a little movie for Nereus here. The first semester I did this, I thought I was going to find Nereus in bait shops, and I couldn't, so I gave him a movie to watch if you want to watch a movie. But these are also polychaetes. This is a Pegmatopoma, it's a honeycomb worm. And so, you know, they have these little crowns of tentacles on the head end like this. If you pull one out of that little tube, it looks basically like a Nereus with a big crown of tentacles on one end, but it has the little parapodia and everything. So when you see those worms that are in a little tube, you're just looking at the little feather duster in, the feather duster is the head, right? But the whole worm is actually down in the tube.
What were those guys called again?
They're polychaete worms. They're tube worms would be the common name for them because they build little tubes that they live in.
Are they,
And-
never mind,
Are they what?
never mind. Yeah. It's just a tube worm, that's all it's called?
Tube worms would be the common name for the whole group, yeah. Tube worms. Okay, oligochaetes, this is, now you know these, right? Lumbricus, and this is tubifex, and this is Elostoma. I don't have a picture of Darrow here. And it has these really long CT. And you see the intestinal tract here?
It almost looks like a walrus when we were like.
Does it look like a worm?
I thought it looked like a big caterpillar.
Or kind of like caterpillar. And did you notice he kind of glides because he has cilia on the underside? So he's actually able, he can do the scrunchy thing, but he can kind of do this gliding thing too. The little planarian too, that Dugessi is ciliated on the underside, so it can kind of do this gliding type of motion. And then Hirudinea would be the leeches, yeah, which obviously produce anticoagulants and suck blood, right?
So I got a question.
Okay.
In Annelidea, that's the only things that we need to know is those three, like, I guess.
Oligochaetes, polychaetes, and Hirudinaeans. Those are the only three classes you have to have.
Okay,
And then
no-
just 4 genera of oligochaetes and 1 of polychaetes. So if I asked you, for example, name a Hirudinaean, probably the only thing you'd say would be leech.
Leeches.
But that's
So
good enough.
leeches and polychaetes only have one in each one, right?
Leech and polychaete only have, well, Hirudinaea and polychaete only have one thing. Polychaete, you probably could say tube worm, or you could say Nereus, right?
Darius and tube worms, that's it?
Or you guys are concerned, that would be it. I guess you'd say sea mouse. Remember that. Okay, let's get a little head start. This was right where I got to the other class, but since I have about 8 minutes with you because I got the moves to work slightly faster, this will be good because most of you that are in Tuesday lab, I want to make sure on Tuesday night that I really get through arthropods, so by the time we go upstairs, you've already seen it. So let's get a little bit through mollusks here. This is it, right? This is if we get through this, we'd be caught up. So the name mollusca means soft-bodied, and this is usually soft-bodied with a shell of some kind, right? Notice these first 3 lines, just like I said, are exactly the same as they were for annelids, right? They're bilaterally symmetrical, and they're triploblastic, and they have an open gut. None of that's particularly special, right? They're protostomes. They're Lophotrochozoan protostomes. They have a true coelom that develops from the splitting of mesodermal tissue, so they have a schistosele, and they have the same type of spiral determinate cleavage that we see in platyhelminths and annelids. You're not going to see anybody with spiral cleavage after this, right? Because when we do nematodes and arthropods, they're going to have idiosyncratic determinate cleavage, and when we get to deuterostomes, they all have radial cleavage. So this is the last time you're going to see spiral cleavage. So you got to really tie it to those 3 groups:
So it's
platyhelminths, annelids, and mollusks. That's it. As far as what we're doing.
What was the annelids, mollusks, and what was the other one?
Platyhelminths,
Platyhelminths.
platforms.
Spiral cleavage?
Yeah. And notice here we have a trochophore larva, just like what we had in annelids. You're not going to see that anymore either, right? Nematodes, arthropods, and deuterostomes, none of them have a trochophore larva. So, but these are the local trochozoa, right? So it makes sense that you're not going to see trochophore larvae after this. Soft body with a shell. 3-part body plan. Head, foot that has the sensory and muscular organs. Visceral mass, which is where the coelom with the internal organs is located. And the mantle, which is the tissue that secretes the shell, right? You saw it on the lining of the clam, or the actual outside part of the squid is the mantle that's secreting the pin. You all remember the ctenidium from the squid, right? It looked like a little feather. Gill, the word gill doesn't really mean much. It just means you have an organ that facilitates gas exchange between blood and water. What do you call an organ that facilitates gas exchange between blood and air? Lung, right? So, but all things we call gills are not homologous to one another. The very specific type of a gill that a mollusk has, that you're going to find in a snail or a clam or a squid, is called a ctenidium. So you'd make me really happy if you could use the word ctenidium. Like on the practical, if I ask you what's this called, can you not say gill? Because I'm going to be forced to take that as right, but I'm going to not like it very much. And a radula, you all know what this is now, right? What's a radula? It's a little tongue with teeth on it that's made out of chitin, right? You saw it in the squid, you saw it in the snail. That is a uniquely molluscan character, right? You don't find a radula in an arthropod or something, or a flatworm. There is no such thing. A radula belongs to mollusks.
All mollusks have that?
Well, remember, like, not all mollusks. So, for example, in the case of bivalves, things like clams, they've
Probably
lost
just
the
lost
radula.
it.
It's lost, right? Because they're highly specialized as filter feeders, so they no longer have a radula. But it is presumably the ancestral mollusk had a radula. You'll see in gastropods, in snails and slugs, that they undergo this very interesting developmental process called torsion. So if you think about it, like things that have big shells like clams and stuff, clams don't like get up and go walking around, right? They tend to just stay buried in sediments or on the bottom somewhere. The only thing with like a really big shell that kind of swims around is nautilus, and of course nautilus has the chambers in the shell that give it buoyancy so that it's able to float. But if you've got a big heavy shell, probably what you're going to want to do if you want to be really mobile, something like a squid, is you're going to highly reduce that shell, bring it inside so that it's just that little remnant pin, and then it doesn't get in your way and you can move around. But if you want to keep a pretty big shell and you want to move around, you got to do something kind of like what snails did. So you know what snails do with their shell, don't you? As they go through development, they twist it. They go through this helical coiling of the shell, and then they throw it on their back like a little backpack so they can walk along. Yeah. So this is characteristic of snails and slugs.
And they don't move their shell, they like, that's their shell.
That's
just
their,
kind
yeah,
of
no,
crap.
right, they don't switch shells. That's a hermit crab does that. Hermit crab doesn't really have a shell, it just goes steal somebody else's shell. But yeah, if you pluck a snail's shell off, that's the end of that snail. He's not going to go find it, not going to go, oh, I'll just go find another shell. It's part of his body, he's not going to be able to be removed from it. The sexes are typically separate in bivalves and cephalopods. Remember you saw the cuttlefish and the squid mating, and there were males and females. In a lot of the gastropods, though, like most land snails, are actually hermaphroditic, right? So you have both male and female in the same animal. And notice, interestingly, in cephalopods, you have a closed circulatory system the way you do in annelids. I don't know of any other invertebrate group besides cephalopods and annelids that have a closed circulatory system. Everybody else is open. What's that a picture of? That's the radula, like a snail, right? You see all those little teeth lined up on the radula. If you asked me what did the last common ancestor to all mollusks look like, we guess it looked maybe something like this. It would have had the 3 basic body parts, right? This sort of head, foot, the visceral mass, which would be where the coelom is that contains the internal organs, and then the mantle that's secreting the shell, right? You notice it would have a radula in its mouth, probably used for feeding. Ventral nerve cords, dorsal circulatory system, right? Just exactly the way that I said it for the cricket or for the worm. You'll notice that it has the gill, which you would call a what? Ctenidium, and you notice that it's posterior. You also notice that it has a one-way digestive tract, right, with a mouth and an anus, which would have been the blastophore, the mouth of the anus. Here's where you have to put it in phylogenetically. Where did it go? You can't tell from this picture, obviously, right? Which would have been the blastophore, the mouth of the anus. These animals are?
Blastophore the mouth.
It would have been the mouth, right? Because these are protostome animals. So this would have been the blastophore here. This developed secondarily. And notice its anus is in the back here so that it poops out to the back end, not on top of its head. You'll see when these snails undergo torsion, what it does is it actually brings the gill to the front, it brings the anus to the front, so they do sleep on their head. That's not particularly great, but that's what they do. Okay, so that would probably be maybe the ancestral form for all mollusks. Notice here is a veliger larva that's a little bit further along, but these definitely go through a trochophore larva stage, very much like an annelid would. This shows you the process of torsion. So this is the way a gastropod would develop without undergoing torsion. And notice that the ctenidia are to the back here, right? The anus is to the back here, right? And undergoing torsion, what it does is it brings the anus and the gill, or the ctenidium, up over the head. That large group of marine snails that includes everything from whelks to limpets to abalone to conchs are called prosobranch. What do you think prosobranch means? Gill forward. Refers to this feature here, right? And the nudibranchs are in a group called opistobranch. What do you think opisto means? Gill to the back. Because guess what they do? This is so ingrained in gastropod development. The gastropods all undergo torsion during development. Opistobranchs undergo detorsion and undo it. But it's such an ingrained feature in them that developmentally they all undergo torsion at some point, even if they undo it later on. So their little gill, their little nudibranch is where? Not prosobranch, but it's opistobranch. It's actually in the back. So these are the 4 classes. I'm going to briefly mention Polyplecophorans. These are the ones that you need to know, right? Because you saw them in lab. Gastropods, snails and slugs, bivalves, which would be anything with 2 shells, and cephalopods, which you dissected one. So this is going to take about 15 minutes at the beginning on Tuesday. Can you not forget how far I got? I'm going to be right here. So we're going to barrel through, and I'm going to finish up mollusks, and then I'm going to rapidly go through nematodes and arthropods, because that's what you're going to see in lab. I'm really determined I'm going to get all the way through them, or very close, so that then we'll just go right upstairs and you'll get to see them all in lab, yeah? And I won't have to talk in lab. I can just let you start the lab, because I'll have done it all down here. Yeah.